Hello students, welcome to the e-learning session. Title of the unit, Free Living Nitrogen Fixing Microbes. Module name, Azospirillum as Biofertilizer. I am Harshala Garg, Assistant Professor, St. Xavier's College, Mapsa, Goa. In this module, you all will be learning about identification of Azospirillum, isolation of Azospirillum, mass multiplication of Azospirillum, production of carrier-based inoculants, techniques of field application and crop response. At the end of module, you all will learn to identify Azospirillum and also learn the methods of isolation, mass multiplication of Azospirillum, methodology of producing carrier-based inoculants, and techniques of field application and crop response. Bajring in 1952 for the first time described a nitrogen fixing bacterium found in the roots of grass species in Brazil and named it as Spirillum lipoferum. Later, Terrard and co-workers in 1978 renamed spirillum as Azospirillum, which means nitrogen fixing spirillum. Azospirillum is found widely occurring in the roots of grass species. Azospirillum lipoferum and Azospirillum braziliensis are commonly used as biofertilizers. How to identify Azospirillum? Azospirillum is a gram-negative, microaerobic, non-fermentive, nitrogen-fixing bacterial genus from the family Rhodospirilleaceae that promotes plant growth. Azospirillum is a motile, vibrioid bacterium. It may possess long polar flagellum and occasionally with peritrichous flagella. It consists of poly-beta hydroxybutyrate granules. Azospirillum behaves as highly aerobic when grown in ammonium-containing medium and found as microaerophilic when grown in nitrogen-free medium. Soil pH also governs the distribution of Azospirillum. The procedure of isolation of Azospirillum goes as further. Azospirillum can be isolated from plant root as well as soil samples. For isolation from plant roots, a suitable host plant is selected. Roots are washed with tap water and cut into 0.5 cm to 1 cm long pieces and sterilized with 0.1% of mercury chloride solution for 1 minute. Sterilized roots are washed with sterile distilled water to remove the sterilant from the surface of the roots. 1 to 2 pieces of roots are placed on sterile and cool semi-solid agar medium containing sodium malate in screw cap tubes and are incubated at 28 to 30 degrees Celsius for 3 to 5 days. After incubation period, at 1 to 2 cm below from the surface of medium, white pellicles of Azospirillum can be observed. The nitrogen-free malate medium becomes blue in color, which indicates the presence of Azospirillum. Once you get Azospirillum in pure culture, one can go for mass multiplication of Azospirillum, which can be used in the production of biofertilizer. In order to produce Azospirillum on mass scale, one needs to procure starter culture. So, starter culture of Azospirillum is prepared by transferring loopful colony to ammonium chloride containing Ocon's medium in culture flask and maintained at room temperature. After 3 days of incubation on rotary shaker, which provides aerobic condition, cells are harvested for inoculation of Ocon's medium filled in fermenter at the rate of 1% of total volume of medium. Optimum temperature of 35 degrees Celsius and sufficient aerobic conditions are maintained in fermenter for few days. At certain intervals, broth is tested for purity and cell number. The broth inoculum is harvested when cell number reaches to 10 raised to power 9 per ml. Production of carrier-based inoculants. In order to produce carrier-based inoculants of Azospirillum, one needs Carriers. Several low-cost and locally available carriers such as farmyard manure, soil, charcoal, peat, etc. can be used. These carriers can be used alone or in combination. It is proved that sterilized soil along with farmyard manure in the ratio of 1:3 proves best carrier for Azospirillum. The bacterial cells survive in farmyard manure for more than 6 months and restores about 10⁶ cells per gram of carrier material. The harvested broth inoculum is mixed with farmyard manure and soil carrier till 40% moisture is obtained or retained. Carrier-based inoculant is packed in polythene bags and the bags are stored at 4 degrees Celsius for about 7 days for further use, or it can be utilized for sale. Coming to the techniques of field application and crop response. Azospirillum inoculant can be applied either as seed treatment, seedling treatment or top dressing. Coming to the first one, that is seed treatment. A slurry of Azospirillum inoculant is prepared by mixing with water in a container. Seeds to be sown in field are soaked in slurry at the rate of 2 kg of inoculant per hectare overnight. Then the seeds are sown in the field in the next morning and the remaining slurry is sprayed in the field. Coming to the second method, that is seedling treatment. In this, slurry of Azospirillum inoculant is prepared by mixing 1 kg of inoculant with 40 liters of water. The roots of seedling to be transplanted are dipped in slurry for 15 to 20 minutes. Then seedlings are transplanted in field. The remaining slurry is sprayed in the field. Coming to the third method of application, that is top dressing. In this method, 3 kg of Azospirillum inoculant is mixed with 25 kg of farmyard manure and 25 kg of soil. This mixture is broadcasted throughout the field by top dressing. Top dressing is applying the inoculant in the form of layer in the field or over crop. Azospirillum inoculant has been widely used in crop production. Various crop responses have been reported from all over the world. Azospirillum benefits the plant growth and increases the crop yield by improving root development and mineral uptake. Apart from nitrogen fixation, growth promoting substance production such as indole acetic acid, disease resistant and drought tolerance are some of the additional benefits of inoculation with Azospirillum. When applied to the sugarcane crop, Azospirillum can save about 25% of recommended dose of nitrogen fertilizer. The experiment conducted in different places in India have indicated the positive response of rice cultivars to Azospirillum inoculants. Experiments conducted at International Rice Research Institute in Philippines showed that inoculation with Azospirillum lipoferum promoted early tillering, growth of rice and significant increase in the filling rate of grain per plant at harvest. Beneficial effects of Azospirillum inoculant have been reported on maize, sorghum, sunflower, cotton and pearl millet. These are the references for further reading. Thank you.